Welcome back to the chapter, my students. And in this module, we shall be studying gene amplification and the first part of polymerase chain reaction or PCR. Now let us move ahead and take a look at what is gene amplification. Now gene amplification is the process of obtaining multiple copies of a known DNA sequence that contains a gene. So in gene amplification, we obtain multiple copies of a DNA sequence. So that becomes the definition of gene amplification. Remember that very well, students. Now let us move ahead and take a look at the process which is used for gene amplification. So here I have polymerase chain reaction or PCR, and PCR is nothing but an in vitro technique of gene amplification. It is also a scientific technique used in molecular biology. And what is it used for? It is used to generate billions of copies of a particular DNA sequence in short time. So that is PCR. Let us move ahead and take a look at brief history of PCR. Here I have a scientist called Gary Mullis, and Gary Mullis has been credited with the discovery of PCR in the year 1983, for which he was also awarded the Nobel Prize in Chemistry in the year 1993. So that was history about PCR. Now moving ahead, we shall take a look at the requirements for PCR. We are amplifying a DNA, and hence my primary requirement becomes the DNA segment. So here I have a DNA segment which is about 100 to 35,000 base pairs long. Now the next requirement becomes primers. Now primers are short oligonucleotide sequences which are about 17 to 30 nucleotides, and they are synthetically prepared as per the requirement. Now my next requirement is DNTPs. DNTPs are deoxyribonucleotide triphosphates. So here I have DATP, DGTP, DTTP and DCTP. So these are the DNTPs that are required. The next is thermostable DNA polymerase. So this thermostable DNA polymerase will be utilized for polymerization of the DNA molecules. And now the final and most important requirement is the PCR machine. Now this PCR machine is an automated machine in which PCR is carried out. It works on the basis of the principle of thermal cycling, which is alternate heating and cooling. So that is the requirement for PCR. Now moving ahead, we shall take a look at the process of PCR. So we begin with what? We begin with a DNA molecule. So here I have a double-stranded DNA molecule. Now I need to amplify it. So let us begin with the process. The first step in amplification is heat denaturation, which is carried out at 91 degrees Celsius. Now heat denaturation will cause the breaking of hydrogen bonds to give me single-stranded DNA molecules. So the double-stranded DNA molecule that was held together by hydrogen bonds will be broken down at 91 degrees Celsius, and I will get single-stranded DNA. Now the next step is annealing. But annealing of what? Annealing of the primers. Now the forward and reverse primers will be paired at the 3' ends of the single-stranded DNA, and this step is carried out at 55 degrees Celsius. So here I have the forward primer and here I have the reverse primer. Now these are the ones which will help in amplification. How? That will take place in the next step, which is termed as polymerization, and it is carried out at 72 degrees Celsius. But polymerization of what? Polymerization of these two primers. But who will carry it out? It will be carried out by a thermostable DNA polymerase called Taq polymerase. How will it do it? It will do it by adding of DNTPs behind the primers. Now Taq polymerase comes from Thermus aquaticus and hence it is called as Taq polymerase. Now here I have Taq polymerase and it is adding the DNTPs behind the primers. So from one DNA molecule I got two new double-stranded DNA molecules. So that is the process of PCR, and the steps are heat denaturation, annealing and polymerization carried out at 91 degrees Celsius, 55 degrees Celsius and 72 degrees Celsius respectively. Remember those temperatures very well, my students. So in this module, we studied gene amplification and the first part of polymerase chain reaction or PCR. And in the next module, we shall continue with polymerase chain reaction or PCR. So till then, study well, take care and bye-bye.